beneath the pale soil of Central Europe, where wheat fields now grow in long unbroken rows above the bones of people who have been forgotten for 5,000 years. There are graves that do not look like any graves that came before them. They are not the careful, deliberate resting places of the Neolithic world, the single burials, the arranged limbs, the offerings of flint and clay and ochre placed beside the dead with tenderness and intention. These are something else entirely. They are mass graves, hastily filled, layered with the bodies of men and women and children who were lowered into the earth, not with ceremony, but with the desperate efficiency of people who had run out of time for grief. They are the first evidence, written in calcium and silence, of what we now understand to have been the first pandemic in recorded human prehistory, an event so catastrophic, so total in its reach, that it did not merely kill a percentage of the population it encountered, but effectively erased entire communities from the landscape of the ancient world, replacing them with a silence that lasted centuries and left echoes that we are only now, with the tools of the 21st century, beginning to understand. Tonight, as the world grows quiet around you and the weight of the day begins to lift from your body, we will walk slowly together through that silence. We will follow the scientists into their filtered rooms and the archaeologists into their careful excavations. And we will try to understand what happened in the Neolithic world of Europe between roughly 3000 and 2500 BCE. What invisible force moved through the settlements and the farmsteads and the longhouses of our ancestors, and what it left behind when it had finished its work. This is a story about death, but it is also, in the deepest sense, a story about us, about the long, unbroken chain of survival that connects those ancient farming communities to the people lying in their beds tonight, breathing the same night air, held by the same gravity, turning under the same slow stars. Let the quiet of the night settle around you like soil settling over old stones. Let your breath slow to match the rhythm of deep, unhurried time. And let the first pandemic tell you its story, gently, in the dark. The graves were found gradually, the way most important things are found, not in a single dramatic moment of revelation, but across decades of patient, often unglamorous work, the accumulation of small discoveries that did not individually seem to mean very much. An archaeologist in Sweden in the early 20th century, digging through what appeared to be a standard Neolithic settlement site, noted with some puzzlement that the burial patterns were irregular. Bodies, placed without the usual care, crowded together in ways that suggested haste rather than ritual. A site in Germany yielded similar anomalies in the 1950s, a site in France in the 1980s, a site in Spain, a site in Britain, each one individually explainable as a local aberration, a community disrupted by some localized event. But taken together, over the span of a century of European archaeology, the pattern that emerged was impossible to explain away as coincidence. Something had happened across the entirety of Neolithic Europe in a relatively compressed period of time, and it had happened not gradually or differently from place to place, but with a terrible consistency. The same hasty burials, the same absence of personal grave goods, the same demographic profile of the dead, the old, and the very young, disproportionate, represented, as they always are, when an infectious disease moves through a population that has no experience of it and no defenses against it. What was perhaps most striking about these sites to the archaeologists who eventually began to consider them as a group rather than as individual anomalies was not the evidence of death itself, but the evidence of what came after the death. In a normal Neolithic settlement, when you excavate the layers carefully and read the stratigraphy, the sequence of soil and debris and artifact that accumulates over centuries of continuous habitation, you find an unbroken record of human presence, tools lost and replaced, pottery broken and discarded, hearth fires rebuilt in the same locations across generations, animals butchered, meals prepared, children born and raised and eventually themselves buried. The record of a living community has a texture to it, a density of material evidence that is unmistakable once you know how to read it. And at site after site across Neolithic Europe, in the layers that correspond to the period between 3000 and 2500 BCE, that texture simply stops. Not gradually, not with the tapering off that might suggest a slow decline or a gradual migration, but abruptly, a hard, clean boundary between habitation and abandonment, between presence and absence, between the world that was and the world that came after. The sites were not destroyed. They were not burned or sacked or flooded. They were simply left. And then, for what the archaeological record suggests were sometimes centuries, they were empty. There is a particular site in what is now Sweden, the site researchers have come to call Frelsegården, though the people who lived there had no such name for it. That illustrates this pattern with unusual clarity. It is not a famous site. It does not appear in popular histories or textbooks. It is one of hundreds of similar sites across the continent, distinguished mainly by the quality of its preservation and the unusual completeness of its stratigraphic record. The people who lived there were farmers of the funnel beaker culture, one of the major agricultural traditions of Northern Europe in the 4th and 3rd millennia BCE. They grew emmer wheat and kept cattle. They made distinctive pottery, wide-mouthed vessels with funnel-shaped necks, decorated with incised lines and impressions that linked them to a network of similar communities, stretching from Scandinavia down through the Baltic and into Central Europe. They built long houses of timber and wattle and daub, oriented carefully with respect to the cardinal directions in ways that suggest a cosmological significance we can no longer fully decode. And then, in the layer that corresponds to approximately 2900 BCE, the site records their absence with the same matter-of-fact completeness with which it had previously recorded their presence. The hearths went cold, the pottery stopped accumulating, the animal bones stopped appearing in the midden. The pollen record, that extraordinarily sensitive archive of past vegetation that collects in lake sediment and peat bogs and can be read with care as a history of landscape change, shows the fields around the site, reverting to scrub and then to woodland over the following centuries, as the cleared land gradually healed itself in the absence of the people who had tended it. It is worth pausing here to consider what this means, not in the abstract language of demographic statistics, but in the concrete, human reality of individual lives. The people who lived at Frelsegården were not abstractions. They were men and women who woke each morning to specific weather, specific light, specific concerns. They had names for each other and for the places they knew, names we will never recover, in a language that left no written record and no living descendants. They had opinions about the best way to set the stakes for a new fence, about the character of the family in the longhouse on the other side of the stream, about the likelihood of rain before the harvest. They had children who needed to be fed and elders who needed to be cared for, and the 1,000 ordinary obligations of community life that fill a day and a life and a generation without ever feeling particularly extraordinary. And then they were gone, not conquered, not converted, not assimilated, gone in the specific, irreversible way that disease-driven extinction is gone, leaving behind only the mute testimony of their abandoned possessions and the bones of their dead in the hastily dug graves at the edge of the settlement. The transition from archaeological mystery to forensic investigation began slowly. In the 1990s, when the first tentative techniques of ancient DNA extraction were being developed and refined, and it accelerated dramatically in the first decades of the 21st century, as those techniques improved beyond anything their original practitioners had imagined possible. The key question for anyone looking at the mass graves and the abandoned settlements was not whether something catastrophic had happened. The evidence for that was overwhelming. But what, specifically, the catastrophic thing had been? Warfare was the first hypothesis, the instinctive resort of minds trained on a historical record in which the collapse of civilizations usually involves armed conflict. But the skeletal evidence did not support it. The bones of the mass grave dead showed no trauma, no cut marks from weapons, no healed or unhealed fractures consistent with violence, no skulls bashed in by the blunt instruments that Neolithic warfare would have employed. The demographic profile was wrong for warfare too. In conflict-driven death assemblages, you tend to see a disproportionate number of young adult males that were warriors and defenders. In the Neolithic mass graves, you see everyone, the old, the young, the male, the female, distributed across the age spectrum in the roughly random way that an infectious disease distributes its victims. When it encounters a population with no prior exposure and therefore no differential immunity, famine was considered and also largely rejected on similar grounds. A famine-driven population collapse tends to produce a more gradual archaeological signature, a slow decline in the density of material culture, evidence of dietary stress in the bones themselves, a pattern of community consolidation as smaller settlements are abandoned in favor of larger ones, where resources can be pooled. The Neolithic collapse showed none of this. It was too fast, too complete, too simultaneous across too wide a geographic area to be explained by crop failure or climate shift, though climate, in the form of a documented cooling period around 3000 BCE, may have played a role in creating the conditions in which something else could unfold with particular ferocity. The something else, as researchers would eventually confirm, was alive, a microorganism so small that it could not be seen with any instrument available to the people it killed, riding silently through the trade networks and the kinship connections that linked the Neolithic communities of Europe into a single, catastrophically vulnerable web. The confirmation came from the teeth. This is perhaps the most extraordinary fact about the story of the first pandemic's discovery, that the key evidence for a catastrophe that reshaped the genetic composition of an entire continent was preserved, not in monuments or artifacts or any of the things we usually think of as the carriers of historical memory, but in the hard mineral matrix of human molars, in the calcified pulp chambers of teeth that had been lying in the soil of Europe for 5,000 years, waiting for someone to develop the tools to read them. The chemistry of it is beautiful in its simplicity. When a living person is infected with a systemic bacterial pathogen, when the bacteria have managed to cross from the gut or the skin or the respiratory tract into the bloodstream itself, the bacteria circulate through the blood, carried by the pumping of the heart to every tissue in the body, including the rich vascular network of the dental pulp. And when that person dies, as so many of the Neolithic mass grave individuals died, acutely and in the full flood of bacteremia, some of those bacterial cells are trapped in the dental pulp at the moment of death. The pulp begins to decay, but the enamel and the dentine do not decay, not on any time scale that matters for our purposes. They are among the hardest and most chemically stable biological materials in nature, and they form a sealed chamber around the pulp, a biological time capsule, as researchers have come to call it, that protects whatever is inside from the contaminating moisture and microbiology of the surrounding soil for periods of time that can extend to tens of thousands of years. The first extractions of ancient pathogen DNA from dental pulp were performed in the late 1990s and early 2000s, using the polymerase chain reaction technology that had transformed molecular biology in the preceding decade. The results were fragmentary and difficult to interpret, and the field was plagued in its early years by the problem of contamination. The risk that modern bacterial DNA, ubiquitous in any laboratory environment, would be amplified along with, or instead of, the ancient sequences the researchers were looking for. The solution to this problem, developed through hard experience over the following 2 decades, was a set of protocols of extraordinary rigor, dedicated clean rooms with positive pressure air filtration, reagents prepared fresh for each extraction, multiple independent replications of every result, the development of bioinformatic methods for distinguishing genuinely ancient DNA, with its characteristic patterns of chemical damage, from the undamaged sequences of modern contaminants. By the 2010s, These protocols had advanced to the point where researchers could reliably extract and sequence DNA from pathogen material preserved in dental pulp from specimens thousands of years old, and the results that began to emerge from the ancient DNA laboratories of Europe were extraordinary. The moment that will stand as the pivot point of this whole story, the moment when the mystery of the Neolithic collapse crossed from the domain of inference and hypothesis into the domain of confirmed fact, came in the form of a sequence of nucleotides assembled on a laboratory screen in Copenhagen in the early morning hours of a winter day in 2015. Though by 2026 we understand it now with a resolution and a completeness that the 2015 researchers could only have dreamed of. The sequence belonged to Yersinia pestis. There it was, not in a medieval grave, not in the body of a victim of the Black Death, not in any context where its presence might have seemed unremarkable, but in the dental pulp of a Neolithic farmer who had died approximately 5,000 years before the medieval pandemic that we had previously considered the earliest known encounter between this organism and the human immune system. The bacterium that would later devastate medieval Europe, that would return again in the 19th and 20th centuries as bubonic plague, that still circulates today in the rodent populations of the American Southwest, Central Asia, and Sub-Saharan Africa. This organism was present in Neolithic Europe 3,000 years before the Black Death. Present, and as the subsequent analysis would make clear, not merely present but active, actively infecting and actively killing the people in whose dental pulp its DNA had been preserved. By 2026, the tools available for this kind of analysis have advanced to a degree that makes the 2015 work look, in retrospect, like a first hesitant step. CRISPR-based sequencing platforms, initially developed for clinical applications in pathogen identification, have been adapted for ancient DNA work in ways that allow researchers to recover viable genetic material from specimens that would have been considered too degraded or too poorly preserved for meaningful analysis just a decade ago. The sensitivity of these systems, their ability to detect and amplify minute quantities of target DNA against a vast background of contaminating material, has effectively extended the temporal reach of ancient pathogen genomics by an order of magnitude. Specimens that the older methods could not read are now yielding sequences of sufficient quality for comparative analysis, phylogenetic placement, and functional inference. And as the database of ancient Yersinia pestis sequences has grown, from the handful of genomes available in 2015 to the hundreds now archived in the international repositories, a picture has emerged of the organism's evolutionary history that is, in its way, as dramatic as any of the events it caused. The picture is this: Yersinia pestis did not arrive in Europe from somewhere else, fully formed and carrying the specific set of genetic capabilities that would make it devastating. It evolved in Europe, or at least in the western Eurasian steppe that bordered Europe's eastern frontier, from a precursor organism, a close relative called Yersinia pseudotuberculosis. That was and remains a relatively minor pathogen of rodents and small mammals, occasionally transmitted to humans through contaminated food or water, capable of causing unpleasant gastrointestinal illness, but not systemic disease, not the sweeping bacteremia and rapid death that its descendant would bring. The evolutionary distance between these two organisms, as measured by the molecular clock that ticks in the accumulation of genetic mutations over time, suggests a divergence date of somewhere between 6,000 and 7,000 years ago, deep in the Neolithic period, long before the first pandemic, long before even the Yamnaya expansion that would carry the organism's later forms westward across the continent. The transformation of a gut pathogen into a systemic killer did not happen all at once. It happened in stages over centuries, driven by the same evolutionary pressures that drive all pathogen evolution. The need to survive, to replicate, to find the next host before the current one recovers or dies. And then there is the gene, the IMNT gene, short for Yersinia murine toxin, a name that reflects the history of its discovery rather than the full scope of its significance. It is a small gene encoding a phospholipase enzyme of modest biochemical complexity. In the context of Yersinia pseudotuberculosis and its relatives, it appears to play a relatively minor role, its function incompletely understood, its loss or gain having no dramatic effect on the organism's basic pathogenic capabilities. But in the context of Yersinia pestis, the YEMT gene is the key to everything, or more precisely, its acquisition is the key to everything. The specific evolutionary step that transformed the organism from a relatively limited pathogen into something capable of continental catastrophe. The reason is biological and elegant, and once you understand it, almost impossible not to find beautiful in a cold and terrible way. Fleas are not passive vehicles. When a flea ingests a blood meal from an infected host, the bacteria it has consumed enter an environment that is profoundly hostile to most organisms. The flea's midgut is acidic, enzymatically active, and rich in compounds that are toxic to the vast majority of bacteria that find themselves there. Most organisms die. The bacteria are digested along with the blood meal, and the transmission chain ends. For Yersinia pestis to become a flea-borne pathogen, for it to acquire the ability to be transmitted from host to host via the flea vector, rather than being limited to the much less efficient route of direct contact or fecal-oral transmission. It needed to survive in the flea's gut long enough to multiply, to form the midgut blockage that is the mechanism by which infected fleas transmit the disease, to wait for the next blood meal and the next host. And the phospholipase enzyme encoded by the IMT gene is precisely what allows it to do this. It protects the bacteria from the oxidative compounds in the flea's midgut, the reactive oxygen species that would otherwise destroy them, by breaking down the phospholipids that are the precursors of those compounds. It is, in the most literal sense, a survival tool, a molecular adaptation to a specific and otherwise lethal environment, and its acquisition, whenever and however it occurred in the evolutionary history of the early pestis lineage, was the moment that changed everything. Before the Yamant gene, Yersinia pestis was a pathogen of limited reach, dangerous to any individual it managed to infect through direct contact, but unable to exploit the vastly more efficient transmission route that fleas provided. After the Yamant gene, it was something else entirely. An organism capable of riding the flea populations that lived in intimate association with the rodent communities of the Eurasian steppe, capable of spilling over into human populations whenever the ecological conditions aligned to bring people and rodents and fleas into sufficient proximity, capable, in the worst circumstances, in the densest and most connected human communities, among populations with no prior exposure and no developed immunity, of moving through a region with the completeness of a fire through dry grass. The reconstruction of the timeline of this mutation, the attempt to date with as much precision as the molecular evidence allows the moment when the YEMT gene was acquired and the subsequent period during which it was being fixed in the pestis population, is one of the more technically demanding achievements of the ancient DNA field, and it is a project that is still ongoing as of 2026. The current best estimate places the acquisition of the YMT gene sometime in the period between 4000 and 3500 BCE, roughly contemporaneous with the early phases of the Yamnaya expansion onto the Pontic-Caspian steppe. This timing is not coincidental. The Yamnaya were pastoral nomads, specialists in the herding of horses and cattle across the vast grasslands north of the Black Sea and the Caspian, and their way of life brought them into constant, intimate contact with the rodent populations of the steppe, the marmots and gerbils and ground squirrels that are, even today, the primary reservoir of Yersinia pestis in Central Asia. If the YEMT gene was acquired during this period, it would have been acquired in the context of a human population that was already mobile, already connected to long-distance trade and kinship networks already poised to carry whatever they encountered across enormous distances, with a speed that the sedentary farming communities of Neolithic Europe could not match. And so the stage was set, not deliberately, not by any agency that intended harm or sought catastrophe, but by the slow, patient, utterly impersonal logic of evolution, by the accumulation of mutations, the selection of variants, the ecological dynamics that brought one organism's newly acquired capabilities into contact with another organism's complete absence of defenses. The Yamnaya began their westward expansion sometime around 3100 BCE. They were impressive people by any measure, tall, by the standards of the time, riding horses in ways that gave them a mobility advantage over any settled population they encountered, bringing with them a Proto-Indo-European language that would eventually give rise to most of the languages spoken in Europe today, bringing bronze metallurgy and new forms of social organization, and, it now appears, a pathogen to which they themselves may have had some degree of partial immunity, accumulated over generations of exposure in the steppe environment, where the organism had been circulating for centuries, and for which the farming peoples of Neolithic Europe had none whatsoever. The Neolithic farmers of Europe had been living in their longhouses and their farming villages for approximately 3,000 years. By the time the Yamnaya and their associated plague lineages arrived, 3,000 years is enough time for a culture to develop deep roots, deep enough that the landscape itself bears their imprint in the cleared fields and the maintained pastures and the accumulated modifications of generations of agricultural work. 3,000 years is enough time for a population to grow dense, for the networks of exchange and kinship and trade that connected community to become rich and complex and far-reaching, and density and connectivity, as every epidemiologist since the 19th century has understood, are precisely the conditions that allow an infectious disease to spread with maximum efficiency. Every connection between communities, a potential transmission route. Every gathering of people at a market or a seasonal festival, a potential amplification event. Every longhouse with its crowded sleeping arrangements and its communal hearth, a petri dish in which a pathogen introduced to one member of the household would find it extraordinarily easy to reach all the others. The demographic estimates that researchers have assembled from the ancient DNA data, from the population genetic analyses that can infer past population sizes and trajectories from the genetic diversity and structure of ancient and modern genomes, are extraordinary in their implications. In the period between approximately 3000 and 2500 BCE, the population of Western Europe appears to have declined by something in the range of 50 to 60%. This is not a figure that lends itself easily to comprehension. A 50 to 60% decline in the human population of a continent, not over 1,000 years, not gradually and replaced as it went, but in what amounts, on an evolutionary timescale, to the blink of an eye, is a catastrophe without precedent in the human story up to that point, and arguably without precedent afterward, until the Black Death of the 14th century, which itself killed perhaps a third to a half of the European population it encountered. The Neolithic collapse was, by this measure, at least as devastating as the Black Death, and possibly more so, in a world where the absolute numbers were smaller and the social and cultural infrastructure for dealing with catastrophic loss was essentially non-existent. It is worth pausing to try to hold this number in the mind, not as an abstraction, but as a reality. If you could stand in a Neolithic farming community in what is now France or Germany or Sweden, around 3000 BCE, and watch the years pass, if you could observe the slow unfolding of the catastrophe from within the community rather than from the vantage point of the archaeological record, what you would see over the span of several decades is the progressive emptying of the world. The neighbors you knew by name, one after another, gone. The elders who remembered the old stories and the old ceremonies, gone before they could pass on what they knew. The children, especially the very young, are always among the most vulnerable when a new pathogen enters a population, and the mass grave data suggests that the youngest members of the Neolithic communities died in disproportionate numbers which meant that not only was the living population being decimated, but the next generation was being taken before it could grow up to replace those lost. This is how populations collapse, not in a single dramatic event, but in a cascade of losses, each one individually survivable, the cumulative weight of them eventually becoming too much to bear. There was a healer. We might call her that, though the word implies a professional specialization that the Neolithic world did not quite have. A woman in one of the farming communities of what is now Denmark, who knew the uses of plants as well as anyone in her community, and better than most. She knew that willow bark, prepared correctly, could reduce a fever. She knew that certain fungi, dried and powdered and mixed with fat, could help a wound resist the rot that otherwise claimed so many injuries. She knew which berries were poison and which were medicine, and in what quantities. The line between the two shifted. She had learned these things from her mother and her mother's grandmother, in the way that practical knowledge had always been transmitted in her world, not through writing, which did not exist, but through the direct transfer of attention from one generation to the next, accumulated across lifetimes too numerous to count, and she was helpless against what was coming. Not through any failure of her own knowledge or skill, but because the thing that was coming was unlike anything her tradition had encountered, and her tradition had, therefore, no knowledge of it to pass on. She treated fevers with her willow bark preparations and watched the fevers continue to rise. She treated the swellings that appeared at the groin and the armpits with the poultices she had been taught, and watched them grow nonetheless. She buried her mother. She buried her apprentice. She buried people she had loved since childhood, and people she had disagreed with, and people she had never quite managed to understand. And she kept going, because the keeping going was the only alternative to the not keeping going, until she too was gone, and with her went a library of botanical knowledge that had taken 30 generations to accumulate and would take no fewer to recover. Him also the trade routes, those remarkable networks of exchange that connected the Neolithic communities of Europe across distances that would have seemed, to anyone living within them, almost impossibly vast. Obsidian from the volcanic islands of the Aegean reached communities in France and Britain. Amber from the Baltic coast appeared in graves along the Mediterranean. Copper tools made from ores mined in the Carpathians showed up in settlements in Scandinavia. These were not accidents. They were the products of sustained, organized systems of exchange that required trust, regularity, and the maintenance of relationships across generations. And they were, in the epidemiological context of the first pandemic, highways. Every trader who carried a bundle of obsidian blades from the Aegean to Cario, Central Europe, potentially carried something else, something invisible, riding in the pulmonary secretions of a mild respiratory illness contracted on the road, or in the blood of a flea that had traveled in the folds of his clothing. The very connectivity that had made the Neolithic world prosperous and culturally rich made it, in the face of a novel pathogen, catastrophically vulnerable. the trade routes that had carried ideas and technologies and beautiful objects across the continent for centuries now, carried death with the same efficiency. By the time the first wave of the pandemic had spent itself, sometime in the mid-3rd millennium BCE, the world that had existed before it effectively gone. Not just demographically, though the demographic loss was staggering and real, but culturally, linguistically, socially. The specific traditions of pottery and house building and ritual practice that had characterized the various regional cultures of Neolithic Europe, the funnel beaker culture, the linear pottery culture, the impressed ware culture, the corded ware culture in its early phases. These traditions did not survive intact into the Bronze Age that followed. They were disrupted, fragmented, in many cases simply ended by the loss of the people who maintained them, the knowledge that lives in practice, the specific gestures of a specific ceramic tradition, the particular words of a particular ritual formula, the technical details of a particular approach to timber frame construction. This kind of knowledge does not survive the death of everyone who holds it, and in communities reduced to 30 or 40% of their former size, the continuity of practice that normally preserves such knowledge was broken in ways that could not be repaired. Into the emptied landscape came the Yamnaya and their successors, the corded ware people, spreading westward from the steppe with a speed and completeness that the ancient DNA record has made astonishing clear. By 2500 BCE, the genetic composition of the European population had been transformed. In Britain, for instance, the ancient DNA evidence suggests that roughly 90% of the genetic ancestry of the population present before the Yamnaya expansion had been replaced within a few centuries, replaced by the genetics of people whose ultimate origins lay in the Pontic-Caspian steppe, thousands of kilometers to the east. This is not a subtle shift. It is, genetically speaking, one of the most dramatic population replacements in the human record, and the fact that it coincided with the period of maximum demographic collapse in the indigenous European population is almost certainly not coincidental. The emptied landscape, the abandoned farms, the unworked fields reverting to woodland, the communities that had once been hubs of regional exchange, now silent and uninhabited, was a landscape that offered minimal resistance to the movement of new people with new animals and new ways of organizing their lives. The Yamnaya did not need to conquer a populated continent. They moved into one that the first pandemic had largely emptied for them. The competing theories about the precise nature of this interaction, about the relative contributions of direct conflict, intermarriage, simple demographic expansion into empty territory, and disease-driven collapse to the eventual outcome, are still being actively debated among researchers, and it would be a simplification to present any single account as definitively established. Some researchers emphasize the evidence for violent conflict in certain sites and regions, the skeletal trauma, the burned settlements, the mass graves that do appear, in a minority of cases, to contain the markers of violent death. Others argue that the scale of the replacement is too large and too geographically uniform to be explained by any mechanism that relies on sustained, violent conflict, and that the epidemiological model, collapse first, expansion second, with little need for overt conquest, better fits the totality of the evidence. Still others suggest that the two processes were simultaneous and mutually reinforcing. That disease weakened the indigenous populations and made whatever conflict did occur more one-sided than it might otherwise have been. Perhaps, as with most great historical transitions, the truth incorporates elements of all these accounts and cannot be fully captured by any of them individually. What the ancient DNA evidence does make definitively clear is the fact of the replacement itself, the genetic transformation of the European population that occurred in the period between 3000 and 2000 BCE. The languages spoken in Europe today, with the exception of Basque and Finnish and a handful of other linguistic isolates, are descended from the Proto-Indo-European language of the Yamnaya and their successors. The genetic ancestry of most contemporary Europeans traces, in substantial proportion, back to the steppe populations that expanded westward in this period. The Bronze Age material culture that replaced the Neolithic traditions of the previous 3 millennia bears the clear imprint of steppe origins in its metallurgy, its animal husbandry, its social organization around warrior elites and the accumulation of prestige goods in every dimension that we can measure: genetic, linguistic, cultural, archaeological. The world of Bronze Age Europe was a fundamentally different world from the Neolithic world that preceded it, and the hinge between those two worlds, the event that created the demographic and social conditions for the transition, was the first pandemic. The scientists who work on this problem in 2026 are, in many ways, the heirs of a tradition that extends back not just to the founding of ancient DNA research in the 1980s, but to all the generations of archaeologists and physical anthropologists who looked at the mass graves and the abandoned settlements and understood, with the intuition of long experience, that something important had happened here, even when they lacked the tools to say precisely what. The CRISPR-based sequencing platforms that are the workhorses of the contemporary ancient DNA laboratory can extract and sequence DNA from samples that would have been completely unreadable by the methods available even 10 years ago. The minute concentrations of surviving genetic material in a fragment of tooth root from a 5,000-year-old mass burial, amplified and assembled into sequences that can be compared base by base with the genomes of every known Yersinia pestis strain ever isolated. The algorithms that manage the bioinformatic analysis of these sequences, that distinguish ancient from modern, that correct for the characteristic patterns of chemical damage in aged DNA, that reconstruct phylogenetic trees from fragmentary and imperfect data, are the products of a decade of intensive development. Borrowing methods from computational biology, machine learning, and statistical physics in combinations that would have seemed fantastical to the pioneers of the field. And what these tools have revealed in the years since the first Neolithic Yersinia pestis genome was published in 2015 is an evolutionary history of extraordinary richness and complexity. The ancient genomes tell a story of a pathogen in the process of becoming itself, acquiring capabilities over centuries and millennia, losing others, navigating the evolutionary trade-offs that confront any organism as it adapts to a new ecological niche. The earliest Neolithic pestis strains, those from the period before or just at the beginning of the pandemic, are in many ways still primitive by the standards of the later, fully evolved pathogen. They lack some of the virulence factors that would make the Black Death strains so devastating. The specific proteins that allow the bacteria to evade certain aspects of the human immune response, the regulatory systems that allow them to switch rapidly between the metabolic states required for survival in the flea gut and the metabolic states required for rapid growth in the warm tissues of a mammalian host. They have the YMT gene, and with it the capacity for flea-borne transmission. But they are, in other respects, a work in progress, an organism still in the early stages of its adaptation to a new set of hosts and a new ecological relationship, still capable of causing devastating disease in a naive population, but not yet equipped with the full arsenal that its descendants would eventually deploy. The AI modeling work that has characterized the field in the mid-2020s has opened new dimensions in the analysis of these ancient genomes, dimensions that extend beyond the historical and evolutionary questions into the explicitly predictive. The attempt to use what we know about the first pandemic to anticipate the next one. The models work by integrating the ancient genomic data with everything we know about the ecology of contemporary Yersinia pestis populations, the environmental and climatic factors that drive spillover events from the rodent reservoir into human populations, the social and demographic conditions that determine how far and how fast a spillover event can spread once it has occurred. They are not crystal balls. The uncertainties in any model of this kind are large enough to humble the most ambitious researcher, but they are tools of unprecedented power for identifying the specific genetic configurations that correlate with increased human risk, for flagging in the vast genetic diversity of the contemporary pathogen population, the variants and combinations that most closely resemble the configurations present in the ancient strains at the moment of their greatest lethality. This work has already produced results of direct practical significance. The identification of specific virulence gene combinations in contemporary Central Asian Yersinia pestis isolates that had not previously been considered high priority for surveillance, prompted by their similarity to ancient high lethality configurations identified in the Neolithic genomic data, led to the initiation of new monitoring programs in those regions. The development of improved diagnostic tools specifically to detect high-priority configurations, rapidly and cheaply enough for deployment in the field settings where rodent plague surveillance is conducted. the initiation of preclinical work on therapeutic targets identified through the comparative analysis of ancient and modern virulence sets, gene sets. None of this means that the next pandemic is being prevented. Pandemics are, by their nature, difficult to prevent, dependent as they are on the accumulation of ecological and evolutionary factors that no human monitoring system can fully track. But the first pandemic is, in this sense, paying forward into the future. The investment that researchers have made in understanding what happened 5,000 years ago is generating, in the form of these practical tools, a dividend that may matter for what happens 5 years or 50 years from now. There is a laboratory technician, a young woman in her late 20s, working the night shift at a genomics institute in Copenhagen, who has spent the past 2 years extracting ancient DNA from Neolithic tooth samples. The work is painstaking and solitary, conducted in a positive pressure clean room where she enters through an airlock and wears full protective clothing, not to protect herself from the samples, but to protect the samples from her. The DNA she is looking for is present in vanishingly small quantities, damaged by 5,000 years of chemical degradation into fragments, sometimes only 20 or 30 base pairs long, indistinguishable to the naked eye from the background noise of contamination that she must learn to read around and through. She has never found the specific Yersinia pestis sequence that would place a new sample definitively in the pandemic period. But she has come close 3 times, and the closeness has given her a feel for the data that she trusts more than she can quite articulate. She works because the work matters, because the answers that are forming in the databases she contributes to will be used by people. She will never meet to make decisions about pathogens she has never encountered, in futures she cannot predict. This is, she has come to understand, the specific dignity of her kind of work, not the drama of discovery, but the patient, cumulative contribution of careful, unremarkable craft to an enterprise that is larger than any individual's portion of it. And so we return, as we must return, to the question that underlies all of this, the question not of what happened, but of what it means. The first pandemic is, in one sense, a story about a specific organism and a specific population and a specific set of ecological and evolutionary circumstances that produced, in the 3rd millennium BCE, a catastrophe of continental scale. In another sense, it is a story about us, about the condition of being human in a world that contains, within the same soil we walk on and the same air we breathe, organisms whose evolutionary interests are not aligned with ours and whose capacity for change is far greater than our own. Every pandemic in the human record, from the plague of Athens to the Spanish flu to COVID-19, has been a variation on this same theme. The encounter between a pathogen and a human population in which the pathogen holds the immediate advantage of novelty, and the human population must improvise its response with whatever immunological and social and technological resources it happens to have available at the time. What changes across the millennia is the nature of those resources, not the fundamental vulnerability which remains. but the capacity to detect and characterize and respond to the threat before it reaches its maximum potential for harm. The Neolithic farmers of Europe had, in this regard, nothing. No awareness that the thing killing them was a living organism. No concept of contagion as a mechanism of transmission. No possibility of quarantine, of antimicrobial treatment, of vaccine-driven preemptive immunity. They had only the biological resources that 5,000 years of human pathogen coevolution had given them, and those resources, shaped by exposure to entirely different pathogens in entirely different ecological settings, were of essentially no use against Yersinia pestis in the initial encounter. They died in the numbers they died in, not because they were weak or primitive or in any way inferior to the people who eventually replaced them, but because they were the first to encounter this particular evolved combination of capabilities without any of the information that subsequent encounters would generate and preserve. They paid, in the most complete way imaginable, the price of priority. We are not, in the deepest sense, so different. The tools available to us are incomparably more powerful. The genomic sequencing, the computational modeling, the global surveillance networks, the pharmaceutical manufacturing capacity, the institutional memory of previous pandemic responses, encoded in protocols and stockpiles and emergency plans. But the fundamental dynamic is unchanged. Somewhere in the rodent populations of Central Asia or Sub-Saharan Africa or the American Southwest, Yersinia pestis is continuing to evolve, accumulating mutations at the slow, relentless pace that is the bacterial version of time. Some of those mutations will be neutral, neither helpful nor harmful to the organism's survival. Some will be deleterious, reducing fitness in ways that natural selection will eventually eliminate, and some, a fraction, small but non-zero, will be in the direction of increased virulence, increased transmissibility, increased capacity to survive in environments that the current versions of the pathogen find difficult. Whether any of those mutations will combine with the others in the specific configuration that would make them dangerous to human populations at scale is not something anyone can predict. It is a question of probability and ecology and the intricate, incalculable play of evolutionary contingency. But the lesson of the Neolithic, the lesson that the first pandemic teaches with the authority of 5,000 years of evidence, is that these questions deserve to be asked and answered before rather than after the event that makes their answers urgent. One of the most unexpected dimensions of the genomic research has been the discovery of traces of ancient immune response in the DNA of modern European populations. When populations survive sustained epidemic pressure over generations, the genetic variants that confer even modest resistance to the causative pathogen tend to be preserved and amplified by natural selection, because individuals carrying those variants are slightly more likely to survive and reproduce than those who do not. This process is slow by human standards, operating over dozens of generations before its effects become statistically visible, but it is consistent and it is detectable in the genomic record if the selection pressure was strong enough to leave a clear signature. And in the case of the first pandemic, with its evidence of devastating and prolonged population impact, researchers have begun to find exactly this kind of signature in the genomes of people living in the regions most severely affected. The variants in question are clustered around the genes that govern the immune response to bacterial pathogens, the genes that control the production of cytokines and encode the surface receptors through which immune cells recognize and respond to foreign invaders. Some of these variants appear in certain frequencies in the genomes of modern populations in ways that correlate loosely with the geographic distribution of the Neolithic collapse. They may be among the earliest examples of pandemic-driven immune evolution in the human genome, a biological memory encoded in the DNA of living people, of an event that no other kind of record preserved. There is another dimension of the legacy of the first pandemic that is less discussed in the scientific literature, but no less real. The dimension of myth and memory, of the ways in which catastrophic events leave traces not only in the biological record, but in the stories that cultures tell about themselves and about the world. The peoples of Neolithic Europe did not have writing, and so they left no direct textual record of what happened to them. But the cultures that succeeded them, and the cultures that succeeded those in turn, did eventually develop writing, and some of what they wrote down may carry the distant echo, however distorted and reworked, of events that were already ancient by the time anyone thought to record them. Some researchers have proposed, though cautiously, that the widespread myths about ages of plague and pestilence found across early literate cultures, about the decimation of earlier peoples by invisible forces, about the world beginning anew after a great dying, may carry deep historical roots in exactly this kind of memory. The memory of catastrophe is one of the things that makes us human, one of the things that our storytelling evolved, in part, to carry. And a catastrophe of this magnitude would have left impressions on the collective imagination of the cultures that formed in its wake that may not be entirely gone. There was a man, one of the last craftsmen of a particular Neolithic tradition of ceramic work in what is now central Germany, who continued making his pots long after the community that had commissioned them was mostly gone. The tradition involved a specific style of incised decoration, geometric patterns pressed into the clay before firing, passed down through a chain of teaching that reached back generations further than anyone could trace. He had learned it from his mother's sister, who had learned it from her own teacher, and so on backward into a past that was not history, but simply the way things had always been. The patterns were not arbitrary. Each element had a meaning, not a meaning that could be translated into words exactly, but a meaning of the kind that resides in the body rather than the mind, the kind of meaning that lives in the gesture of making and is lost when the making stops. He continued because the making was what he knew, and because somewhere in the hope of making, he preserved the possibility, however diminishing, that there would be someone to receive what he made. The pots that survived him sit in museum cases and receive the attention of researchers who study them for what they reveal about Neolithic technical traditions. The man who made them receives no attention at all because there is no way to know that he existed. And yet he did, and the pots are his. And the fact that they survived when everything else about his world did not is its own kind of quiet miracle, the kind that history produces without intending to and without anyone to notice. And so we are left with this. The story of the first pandemic is not a story that ends. Not with the abandonment of the last Neolithic farmstead, not with the arrival of the first Yamnaya horsemen on the Western European plain, not with the reconstruction of the first ancient Yersinia pestis genome on a laboratory screen, not even with the deployment of the AI models and the surveillance programs and the improved therapeutics that the ancient DNA research is currently generating. It is a story that continues in every dimension: biological, historical, scientific, and deeply, personally human, because the organism continues, and because we continue, and because the relationship between organisms and the human immune systems that must respond to them is not a problem that admits of final solution, but only of ongoing, vigilant, imperfect management. The first pandemic is not a cautionary tale, not exactly, though it contains cautions that are urgent and real. It is something larger than that, a founding event, in a sense, of the world as we know it, the catastrophic opening of a chapter that has not yet closed. Every epidemic that followed it, the plague of Athens, the Antonine Plague, the plague of Justinian, the Black Death, the 1918 influenza, COVID-19, and the outbreaks whose names have not yet been given because they have not yet occurred. Every one of these events was, in a sense, a re-encounter with the same fundamental dynamic, the same relationship between pathogen evolution and host vulnerability that the Neolithic world encountered first and survived, barely, in ways that shaped everything that came after. We are the inheritors of that survival, the specific, irreplaceable fact of our presence in the world tonight, breathing, warm, alive, is the downstream consequence of the survival of people who had no reason to expect that survival and no understanding of what they were surviving. They kept going because the keeping going was the only available alternative. And here, across 5,000 years of unbroken biological continuity, we are. Tonight, as your breathing slows and the weight of the day releases its hold on your body, the soil of Central Europe continues its ancient, patient work, settling, molecule by molecule, over the bones of people who are 5,000 years gone and still, in some small chemical way, here. The sequencing machines, in their clean rooms, continue their reading, assembling sequences from fragments too small to see, sending their findings into the databases where the AI models will find them and build from them the tools that the next generation of scientists will need. And somewhere in a field in Scandinavia, above a mass grave that has not yet been found, the frost is slowly releasing its grip on the topsoil, and the earthworms are beginning, in their slow and essential, to do their work. The story does not end, but you can rest within it now, the way a stone rests within the river, present in the current, held by the water, part of the same long flow that has been moving since before the beginning of the human record and will be moving long after the last of our names is forgotten. You are whoever came next. You are the latest in an unbroken chain of survivors that reaches back past the first pandemic, past all the pandemics, past everything, back to the beginning of the human story, whenever and wherever that was. And tonight you are safe, and the night is quiet, and the world beneath you has held heavier things than worry, and will hold you too, gently and without asking anything in return. The earth turns slowly in its old and patient way. The soil settles. The stars continue their long, indifferent arcs across the sky, above the fields where so much was lost and so much impossibly endured. The night deepens around you. The breath slows. The ancient and the new rest together in the same darkness, held by the same gravity, moving in the same great slow arc. It is worth dwelling, for a moment, on the strangeness of this situation, the extraordinary fact that the most transformative event in European prehistory left no direct record of itself in any form that human beings could read for 5,000 years. The people who experienced it did not write about it. They could not. They had no writing. They did not carve it into stone or paint it onto the walls of caves. They did not, so far as we can tell, build monuments to the dead that would encode in their scale and their permanence some sense of the magnitude of what was lost. They simply died, and the world went on without them, and the evidence they left was the mute evidence of absence, the untended fields, the empty houses, the bones in the Massmers' graves, the pollen record in the lake sediments showing the slow return of the forest to the land they had cleared. This absence of direct record is not merely an obstacle to understanding. It is itself a kind of testimony, a testimony to the completeness of the collapse, to the rapidity and the totality of the loss. A culture that survives a catastrophe tends to leave records of it, because the act of surviving creates the need to explain and commemorate what happened, to give the experience a form that can be transmitted to those who come after. The cultures of the Neolithic did not survive in the form in which they existed before the pandemic. They were not diminished and then recovered. They were, in the regions most severely affected, effectively ended. Not every individual killed, but the social and cultural continuity broken past the point of repair, the chains of transmission by which knowledge and practice and identity move from one generation to the next, severed in too many places at once to be reconnected. What came after was not a continuation of what had come before. It was something new, shaped in part by the memory of the old, preserved in the bones that the new people buried alongside their own, in the pottery styles that were partially adopted and partially transformed, in the ritual practices that left faint but detectable traces in the archaeological record, but fundamentally, irreversibly new. The Bronze Age world of Europe was not the Neolithic world recovered. It was a different world, built on the ruins of the old one by people who were, genetically and culturally largely strangers to the land they came to inhabit. There is a particular kind of melancholy in this fact that does not fully yield to analysis. The Neolithic people of Europe, the farmers and the herders and the craftspeople and the healers, the people who built Stonehenge and the megalithic passage tombs of Ireland, and the causewayed enclosures of Britain, and the lake dwellings of the Alpine valleys. These people were not a failed civilization, not a primitive prelude to something better. They were a fully realized human world, with their own wisdom and their own beauty and their own ways of being in relationship with the land and with each other, ways that had been refined across 3,000 years of agricultural life and that were adapted with extraordinary sophistication to the specific conditions of the specific places they inhabited. The pottery they made was beautiful, genuinely, objectively beautiful, in the way that things made with skill and care and cultural depth are always beautiful, regardless of the technology available. The monuments they built encode a knowledge of astronomy and landscape that we are still working to fully understand. The social arrangements that the archaeological record implies, the patterns of communal labor in monument construction, the evidence of long-distance exchange networks maintained across generations, the care given to the dead in the pre-pandemic burial traditions suggest communities of considerable social complexity and considerable social warmth. All of this was lost, not preserved in a museum, not transmitted in a different form, not absorbed into the cultures that followed and transformed into something recognizable, lost with the finality and the indifference that mass death brings to the accumulated achievements of the generations that preceded it. The potters and the monument builders and the healers and the storytellers and the people who knew which stars meant that it was time to plant the emmer wheat. They are gone in a way that is more complete than the going of most things, because even the cultural memory of their existence, the sense that there had once been a different world here with different people and different ways, did not survive the transition to the Bronze Age in any form we can clearly identify. The Yamnaya and their successors did not know, or did not need to know, or did not choose to remember the specific human achievements of the world that had preceded them in the landscape they came to occupy. They brought their own traditions, their own cosmologies, their own ways of understanding what the world was and how to live in it. And those traditions, those cosmologies, those ways, deeply shaped by the steppe environment that had formed them, by the horse and the wheel and the bronze technology that had made the expansion possible, are the ones that survived into the historical record and gave rise, in their long subsequent transformation, to much of what we are. There is a strange comfort in the genomic evidence, if comfort is the right word, the evidence that modern Europeans carry in their DNA, the genetic legacy of the Neolithic farmers who were so largely displaced, the roughly 10 to 20% of ancestry that traces back to the pre-Yamnaya population means that the Neolithic people of Europe were not completely erased. not even in the most biological sense. They are here, in diluted and transformed form, in the genomes of the people who live today, in the same landscape they farmed 5,000 years ago. The woman who grew the emmer wheat in what is now France. The man who made the pots in what is now Germany. The healer who knew the uses of plants in what is now Denmark. Their genetic contributions, however attenuated and rearranged by 50 generations of intermarriage with the populations that followed them, are present in the people walking those landscapes today. Not recoverable as individuals, not identifiable by any means available to us, but present as a statistical reality in the population-level patterns of genetic variation that the ancient DNA research has now mapped with considerable precision. The dead are not entirely gone. They are here, in a form that would have been incomprehensible to them, encoded in the biology of descendants who do not know their names. The scientific work of reconstructing the first pandemic continues, and it continues in directions that were not imaginable even a decade ago. The ancient DNA databases are growing. Every new excavation of a Neolithic site, every new analysis of previously unsampled skeletal material, adds data points that refine the picture of when and where and how intensively the pathogen was circulating. The phylogenetic analysis of the ancient Yersinia pestis genomes is being integrated with increasing sophistication with the ecological and climate data that is now available for the Neolithic period from sources as diverse as ice cores and pollen records and ancient lake sediments and the growth rings of ancient trees. The AI modeling work is generating hypotheses about transmission routes, about the role of animal domestication in creating the conditions for spillover, about the specific ecological configurations that seem to correlate with the most intense periods of epidemic mortality that are being tested against the archaeological and genomic data in a productive cycle of prediction and verification that is, in the best traditions of science, making each iteration of the model better and more predictive than the last. And the questions that cannot be answered by any of these methods, the questions about experience, about meaning, about the inner life of people who had no way of understanding what was happening to them. These questions remain open, as they always will, held by the imagination rather than by the evidence, resistant to the kind of closure that scientific progress provides. They are held by writers and artists, and by ordinary people who learn about the first pandemic and find themselves stopped, as one always is, by the most important historical facts, by the sheer weight of what it means, the weight of the specific, unrepeatable human lives that are compressed in the archaeological record into the abstraction of demographic percentages and genetic replacement events and phylogenetic trees. It means something, this story, something beyond its scientific interest, beyond its epidemiological relevance, beyond its significance for the deep history of European populations and languages. It means something about what we are, about the specific kind of animal that we are, the kind that can be reduced to 30% of its former numbers, and still, in the survivors, find the will and the capacity to continue, to keep the fires burning and the fields tended and the children fed and the knowledge passed forward, even when the world that had made that knowledge meaningful was effectively gone. It means something about the stubborn, unreasonable persistence of the human desire for continuity, for the sense that what we do and what we know and what we love will outlast us in some form, even when the evidence suggests that it will not. The Neolithic farmers kept farming after the dying began, not because they had any reason to think it would help, but because farming was what they were and what they knew, and because the act of continuing to do the thing that defined them was the only form of protest against the erasure that was available to them. Now, in the warm quiet of wherever you are, with the night settled fully around you, and the day's concerns grown distant and small, let the 5,000 years between you and the first pandemic do their work. Let the distance be not alienating but comforting. The evidence that what happened then, however complete it seemed, was not in fact the end of the story, that the story continued, that you are the proof of its continuation. The farming communities of Neolithic Europe were not the last chapter. They were among the early chapters of something that is still being written, and the writing includes you tonight, in your warmth and your safety and your quiet, reading the story of the people who came before. The soil of Europe is deep with time, every field, every garden, every patch of earth disturbed by a spade or a plough passes through layers of history, the medieval, the Roman, the Iron Age, the Bronze Age, and below all of that, the Neolithic, the first world of European farming, its bones and its broken pots, and its abandoned longhouse timbers composted back into the earth from which the wheat still grows. The first pandemic is in that soil. The bodies of its victims are in that soil. Their calcium returned to the groundwater, their carbon breathed out over centuries into the atmosphere that passes through our lungs tonight, in the most literal biochemical sense, made partly of the same atoms that once made them. The recycling of matter that the universe performs continuously and without sentiment has seen to that. We share not only their history but their substance, distributed and recombined across the millennia into the specific, temporary arrangements of atoms that we call ourselves. Consider, as your eyes grow heavy and the darkness behind them begins to deepen and soften, the remarkable fact of scientific curiosity itself. The fact that there are human beings who have devoted years and decades of their working lives to the project of extracting legible information from the dental pulp of people who died 5,000 years ago. This is not an obvious thing to do with a human life. It does not, in most conventional senses, pay well, or bring fame, or produce the kind of immediate, tangible results that most people associate with meaningful work. It requires extraordinary patience. The patience to spend months developing a technique that may produce no usable data. The patience to repeat an extraction a dozen times before the results are clean enough to be meaningful. The patience to write papers whose implications will take years to be recognized and absorbed by the wider scientific community. it requires a particular kind of faith, not religious faith, but the scientist's faith, which is the faith that reality is knowable and that the patient application of rigorous method will eventually, if imperfectly, reveal it. And it produces, in the end, something that is genuinely extraordinary. The ability to hear, however faintly and indirectly, the voices of people who have been silent for 5 millennia. This is what the ancient DNA researchers have given us. Not certainty, not a complete and final account of what happened in Neolithic Europe and why, but something more valuable and more honest than certainty. The beginning of a conversation with the deep past that was not possible before. A conversation in which the ancient dead are not merely the passive objects of historical inquiry, but active participants, communicating through the medium of their biology, the facts of their lives and their deaths that they could not have communicated in any other way. The healer in Denmark, who watched her patients die, did not intend to leave behind, in the dental pulp of the last patient she treated, the genetic signature of the pathogen that defeated her. But she did, and it is there, and it has been read, and it has changed what we know about the world. This is the specific, strange, and moving beauty of ancient DNA research, the way it allows the most private biological facts of long-dead individuals to become, without their knowledge or consent, but perhaps not entirely, without their interest, evidence in the reconstruction of the history of their world. Let that thought settle gently into the quiet of the night. Let the distance between you and the first pandemic collapse, just for a moment, into the single continuous fact of human persistence, the unbroken line of people who encountered impossible things and survived them, not always intact, not always unchanged, but always as long as we are here surviving. And then let it expand again, back to its full 5,000 years, back to the comfortable vastness of deep time in which individual lives are small and mortality is universal, and the fact of any particular person's survival is both completely unimportant on the cosmic scale and completely, irreducibly precious on the human one. You are here. That is the final fact, and it is enough, more than enough, tonight in the quiet, with the night deepening around you and the world settling into its slow, patient turning. The earth receives the darkness. The soil breathes its quiet breath. Somewhere in a clean room in Copenhagen, a sequencing machine reads another fragment of 5,000-year-old memory and sends it, in the language of molecules, toward its place in the long story of what we are and where we came from. And somewhere in the fields of Central Europe, above the bones of people who kept going for as long as they could and then passed the world on to whoever came next, the night is soft and the stars are out and the wheat is growing in the dark, patient and unhurried, fed by the same earth that holds the first pandemic in its keeping. The night deepens, the breath slows to the rhythm of deep, unhurried time. The ancient and the new rest together in the same darkness, held by the same gravity, moving in the same great slow arc that has no beginning we can find, and no end we can see, and all is still.